Friends, welcome to lecture 2 of Biotechnology. Here I am going to talk about genetic engineering. What is genetic engineering? So, before that, let's start with yesterday's topic bioprocess engineering. I told you that we should maintain a sterile medium for genetic engineering. Why? Why do we need it? Have you ever noticed in home when we boil some milk, sometimes we see that the milk turns into curd or turns into uh, cheese, like I don't know what is the term, but like cheese. So we say the milk is spoiled. So why that happens? We are not adding anything to the milk, because suppose this is a pot, okay, and we have our milk here, but we don't know that in the air and even in the pot or even in the milk there are thousands and crores of bacteria. So there are thousands, millions and billions of microorganisms that we cannot see with our naked eyes. And if we stop boiling or somehow those microbes comes into our milk, then and they if they have the ability to convert it into curd, then it is going to spoil. Okay, so we know that we don't understand that there are many microbes all the time around us. That's why traditional hybridization method often results in inclusion and multiplication of undesirable genes along with desirable genes. You might get your desirable genes, but also you are going to get some undesirable genes, which might cause mutation in your body. Let's give an example, like you are making hemulin, that is insulin, from E. coli, right? So, what if with those peptide chains, some other kind of deformed protein are also, you know, also comes to that, then that protein can cause severe damage to your body. Also, it can cause mutation. That's why we need bioprocess engineering in our biotechnology. So, genetic engineering is mainly okay. Just tell me, what is engineering? What do we know by engineering? Basically, engineering is to make something, to build up something. Like we build, engineers build PC, they build, and the civil engineers, they build bridges, houses. Where the genetic engineers are also going to build something, and that is some special kind of DNA. Now you'd ask me, how can we make DNA? Right? Yeah, we cannot. We cannot make DNA. We are not God. So, but we can change DNA. How? That is called the recombinant DNA. We're going to change DNA. We're going to add something, or we're going to remove something from DNA. That is called recombinant DNA. This is the main topic of biotechnology. Biotechnology rounds up around this only topic, that is rDNA. Via rDNA we can also do gene cloning and gene transfer, and this overcome the limitation and allows inclusion of only desirable gene, because we already have bioprocess engineering and this is genetic engineering. With this we can do rDNA. We can make rDNA. Now we are going to know what is rDNA. This is very important, and I am going to make a little diagram to make you understand about rDNA. It's very easy. Just see. Suppose this is a concerned organism. So what is a consent organism? Suppose this is an organism which does not have s- the ability to make a protein, right? So it cannot make a protein, that's why it is having some kind of disease, and it needs a gene, that a specific gene that would make a protein. Suppose this is its gene, this is the DNA of this consent organism, right? Now it does not have a specific gene that can make a protein. So this gene cannot make a protein. This gene cannot make a protein, but if we add some the gene which can make the protein, then the same organism can make the protein, right? So it is a very very easy funda. So what we are going to do, we are going to take this DNA aside, we are going to take away from that organism, and we are going to add this part, like this gene, to this DNA. But it's not simple, like we are not playing with paper and scissor that we would cut and then we would add with glue, but we have techniques how we are going to add this gene to this DNA. For that we need a vector. I'm going to talk a lot about vector, but not now, later, because it's a whole big topic. So suppose this is a vector. Vectors are usually plasmids. What we are going to do, we are going to add this particular gene with this vector. So this is called a whole vector. Before adding the gene to this vector, it was just a plasmid, like a normal plasmid. And now, like you have to give a message to someone, supposedly a letter, and your brother, and you have a brother, right? So your brother is normally a brother, but if you give your brother that letter and tell him to post it, then he would become a messenger to you, he would become a vector to you. Before having the letter, he was your brother, just your brother, but now he has your letter and he is going to transfer it, so he is a messenger, he is a vector. Like that, if he does not have the desirable gene, then it's just a plastic. But when we are giving the gene inside the vector, then it's called inside the plasmid, then it's called a vector. Now we're going to add them and they are going to become our DNA. And now what we are going to do, we are going to again now we have the ability to make the protein. It is that simple.